Thank you very much for the opportunity to be here. Montreal is one of my favorite cities, and I was particularly pleased to get the opportunity to come. So I don't really need to tell this audience how extraordinarily important viral hepatitis infections are, but with 185 million people and nearly 250,000 Canadians infected. But I think we should really consider the fact that these two viruses together really rival the big killers in infectious disease worldwide, and they deserve significant attention. So the number of people who died of these infections in 2010 is really quite comparable to what we saw in deaths from other important infections like tuberculosis and HIV. So as we know, the past for patients has really represented small incremental steps forward with interferon-based regimens, and these regimens have steadily, over time, increased the response rate slightly. First, the addition of ribavirin, pegylation of interferon, and with the first medications, the first generation protease inhibitors, we did see a significant increase in response rates, but the toxicity of the regimens also increased, and that really is the major limitation to interferon-based therapies. Interferon adversely affects nearly every system of the body, and the driver was really to try to eliminate interferon-based therapies using our scientific understanding of how the virus replicates, and that really was achieved through a number of great basic science advancements in which we learned which proteins are critical to viral replication, and agents were designed, small molecules were designed that could then block the functioning of those critical proteins in replication. So we're now in an era where more than 95% of patients treated with these regimens are responding with sustained virologic response or cure. And this has been a huge advancement that this may seem a little bit irrelevant to how close we are to a vaccine, but I would say that the problem is that it started the notion that hepatitis C is an infection that needn't be considered anymore because we have these highly effective cures and may in fact have set back the development of a hepatitis C vaccine. So there's no doubt that these therapies have improved dramatically, very tolerable therapies with really dramatic cure rates. But I think we still have some challenges. The first challenge is really finding the people who need treatment. And why is this so challenging? Because as we know, hepatitis C is a clinically silent. I don't want to say silent because there are plenty of symptoms. People feel fatigue and other things, but it tends to not present to physicians until end-stage liver disease is present. In addition, knowledge of infection status is very limited, and it's estimated that only about 5% of people in the world who are infected with this virus know it. So you do much better in Canada than in many places in the world. You can see on the x-axis, sorry, I don't really have a way of pointing because there are two screens, but on the x-axis, the rate of diagnosis, and the y-axis is the treatment rates, and the size of the pink bubble is essentially the burden of disease. And so I have to applaud Canada because it definitely stands out, and the countries to the far right, with a larger percent than is typical of people with hepatitis C having been diagnosed. But you can see that there's significant burden of disease in countries where there's actually very low rate of diagnosis and treatment. So an additional challenge is that the highest risk groups for hepatitis C are marginalized groups. They include people who inject drugs, and they include people who live in these highly endemic regions of the world. And the regions of the world where hepatitis C is highly endemic, those individuals tend to have limited access to testing. There's poor needle injection and blood product hygiene, and there's, of course, limited access to therapies. So this map of the world, of the 185 million people who have hepatitis C, shows by color the burden of disease. So red is the highest burden of disease, and the black dots on the country indicate how many people in that country have the infection. So you can see that the large black dots are in regions of the world where medical health care has significant limitations in its delivery. These are areas where it's not possible to even uniformly screen the blood supply, where there's reuse of medical equipment quite frequently, and the notion that we will easily get into those areas and treat patients is a complicated one. So in addition, I'm not going to address cost, but we know that treatment does remain expensive, and although there are drivers to bring that down, it is a problem. There is also has been a dramatic reduction in side effects of these medications, but there do remain some side effects from these medications, particularly when we start using them in larger numbers and combinations. An important issue is, let's just say that we find everyone who's infected and we begin to treat them. There are some additional limitations of these drugs. And among those limitations, the drugs do not provide protection against reinfection. So in this study, in this Norwegian study, they looked at the incidence of hepatitis C virus infection following a sustained virologic response. So they looked at individuals who were treated between 2004 and 5, and they required that if you had a history of injection drug use, that you abstain for at least 6 months before initiating therapy. And they then followed up with those individuals between 2012 and 2014, and they found that there were 94 individuals with people who had a history of injecting drugs, and 44 with no history who had achieved a sustained virologic response. They followed those individuals for around 7 years. And what you see is that there were reinfections in this group, that almost 40% of the people who injected drugs, again, requiring abstinence for at least 6 months before they initiated therapy, relapsed into injection drug use. And of the 12 people in whom they saw reinfection, 11 of them acknowledged that they were actively injecting drugs. And so when you look at the incidence of infection in this study, patients with no history of injection drug use prior to treatment, that's the bar all the way to the right, they had fairly low rates of infection. But in individuals who had a prior history of injection drug use, there was a higher incidence of infection, and the incidence per 100 person years was 4.7 in those who relapsed into injection drug use. So this is relevant not just in people who inject drugs, but in the regions of the world where hepatitis C is endemic. Remember that these individuals will continue to live in that environment and will remain at ongoing risk of exposure to hepatitis C. So this is not just a message of importance in people who inject drugs, but in any setting in which individuals remain at risk of ongoing exposure to the infection. In addition, we've seen reinfection with the DAAs, directly acting antiviral resistant HCV. So beginning with the first clinical trials of these agents, we began to see reinfection. So this is a case that was highlighted in a publication in 2014. There were 2 HIV-infected men who were sexual partners, one of whom was treated successfully and achieved a sustained virologic response. The other actually failed DAA therapy and had a documented telaprevir protease inhibitor resistant mutation. And unfortunately, the partner who had a sustained virologic response became viremic again, and upon sequencing the virus, it was determined that that man had acquired the DAA-resistant hepatitis C virus infection from his sexual partner. So these phylogenetic trees show that the virus in the man who had a sustained virologic response was transmitted by his sexual partner and did have that drug resistance-associated variant. So we don't know the potential impact of DAA resistance yet because there haven't been enough people treated who have not successfully responded. As I mentioned, the treatment success rates are over 95%, so we have not had a lot of individuals fail treatment at this point, but there has been an assessment of what happens in at least a few settings when people do fail, with the question, how long do these resistance variants persist? So in a treatment regimen composed of 3 different classes, an NS3, NS5A, and NS5B inhibitor, they analyzed 2,500 individuals who in phase 2 and 3 trials received that combination, and they were all pulled together. It didn't matter what dose or duration they got, and they looked at all those individuals, and they found there were 67 genotype 1A, and consistent with these drugs being more effective against 1B, only 7 genotype 1B failures. So this is a small percent of the population. 2.9% of the patients failed. But what they found is that when they followed these individuals over years subsequently after discontinuation of therapy, that the NS5A resistance-associated variants persisted for at least 48 weeks after discontinuation of therapy. Similarly, although the NNI mutations could persist, they were rarely found, but they persisted for a long time. So the NS3 resistance-associated variants did decline during that time period and seemed to go away, but at least some of these resistance mutations seemed durable. And what we don't know is how much that will affect treatment as they become more prevalent in the population. But we saw this with HIV, where resistance mutations became more common in the population as we expanded access to treatment, and more individuals were then infected initially with resistance-associated variants in HIV. And so I think we may see this in hepatitis C as well. But again, the impact is unclear. And the last point that I will make is that treatment in the later stages of disease is essentially often too late. So we know that there is enormous benefit to treatment of hepatitis C virus infection and cure. So shown over time is the rate of liver failure, and you can see the curve without sustained virologic response. This is why we treat people. We know that people who have advanced liver disease progress to end-stage liver disease and liver failure if we don't treat them. But if you look at the curve on the bottom with sustained virologic response, there are still some steps upward in that curve. And for those of us who take care of, and these data were generated with interferon-based regimens, but we suspect that the same thing will be true with the DAAs. And for those of us who treat patients with hepatitis C, we do in some cases eradicate hepatitis C, but the patients continue to have disease progression, in some cases liver cancer, and some cases liver failure. So I think an ounce of prevention really continues to be worth a pound of cure. It is far better to prevent people from acquiring hepatitis C virus infection than to try to treat them with these complications continuing to arise if we've waited too long because we didn't find them earlier. So in summary, I think we can look at the 185 million people in the world. A very small proportion of them, it's estimated about 5%, are aware they're infected. Of those, a very small proportion of them are treated. And while the treatment rates are very high, what we have is a very small proportion of the world's hep C infected population being cured of hepatitis C. Further complicating that, a subset of those will continue to have severe liver disease and disease progression, and a subset of them, unknown how many, will become reinfected and essentially re-enter the large box again. So I would argue that I think we need to consider a hepatitis C virus vaccine in combination with these extraordinarily potent and effective drugs, and that likely gives us our best chance for getting control over this infection on a global scale. So if we believe we need a vaccine, the question is it even possible to be protected against hepatitis C infection? So one of the things that made us think that this might not be a possibility is that individuals who've spontaneously controlled hepatitis C virus infection have been documented to be reinfected, and in the cases of reinfection, in some cases, they've become persistently infected. So this really was discouraging to the field and made us feel like it's not even possible to have protective immunity to hepatitis C virus infection. However, we do have some evidence that says there is protective immunity. And in order to introduce this, I'm going to introduce a cohort that I've directed. It's called the Baltimore Before and After Acute Study of Hepatitis. It enrolls young, 18 to 35-year-old, in people who are injecting drugs. At the start of the trial, they need to be antibody, hep C antibody, and virus uninfected, so uninfected. And we follow them over time. Once a month, we screen them for the presence of HCV RNA, which, as you know, appears shortly before the development of antibodies. And there are two outcomes of infection: persistent infection or spontaneously controlling the infection. And so if you look down the left side of this chart, that's the important one. We looked at 113 people who acquired hepatitis C virus infection, and of those, 73% of them became persistently infected, which is what we know. About 3/4 of people infected become persistently infected, and spontaneous control was the less common outcome. But when we looked at the individuals who'd spontaneously controlled infection, Remember, this is in the setting of people with ongoing active injection drug use. And although we counseled them to discontinue this and put them in drug treatment programs, we're not always effective in intervening. And so in some cases, people become reinfected. In fact, in half of the participants in our trial that we followed longitudinally who had controlled infection, we could detect a new infection in them. So by sequencing a different virus. And so the outcome in those individuals, though, is exactly the opposite of people infected initially. So if you look at the bottom two squares, that shows you that essentially only 17% of the people who were reinfected became persistently infected. So it's really the exact opposite of spontaneous control rates in the initial infection, with the vast majority, 83% of people, controlling a second infection. So we know that may be due to something, a fixed genetic trait, for example, something about that person just makes them able to clear Hep C, and it's not an adaptive immune response that we might hope to mimic with vaccination. But actually what we saw in these individuals, if you look at on the y-axis maximum detected HCV RNA levels at either during the primary infection or in subsequent infections, in most cases, the magnitude, the maximum detected HCV RNA was lower in the subsequent infections. So there are a couple exceptions to that, and I'll point out that the two people I've circled here are the two people who became persistently infected. So one of the persons who has a higher maximum HCV RNA was actually someone who went on to become persistently infected. And this is striking because, remember, we're not sampling them at a set time after re-exposure. We're randomly pulling them in on a monthly basis, random basis on the basis of when they were infected, and yet they all have lower maximum levels of HCV RNA with just those two exceptions. So I think we could say that they don't just control Hep C, but they seem to actually do it early with a lower level of maximum virus. The other thing you could ask is the duration of viremia, and that actually also is lower. Now here, of course, I've eliminated the people who become persistently infected, so looking at individuals who clear a primary infection and subsequent infection, the duration of time for which we can detect virus is much shorter. So is this an immunologic response? We also see broadening of T-cell responses in HCV reinfection. So if we look at people who are chronically infected, the development of T-cell responses, new T-cell responses over time is shown here. And after the first few hundred days of infection, it's very rare for people with chronic infection to develop T-cell responses against new T-cell targets or epitopes. In contrast, in people who are reinfected, they continue to develop new responses to new parts of the HCV polyprotein over time. These results have been confirmed in the Montreal acute hepatitis C injection drug user cohort run by Julie Bruno and Nagla Shukri, and they basically also saw an increased magnitude and breadth of T-cell responses in people who were repeatedly exposed and controlled the infection. They also saw higher capacity of T-cells to proliferate in people who controlled the infection, and this was really associated for the immunologists in the group with the expansion of a specific subset of CD8 T-cells, and also with production of at least individual cytokines more readily from T cells in the CD4 population. So what we know in these individuals who repeatedly clear, which again happens in 83% of reinfections, the exact opposite of primary infection, is that we see recurrent infection is associated with this lower maximum HCV RNA level and shorter duration of viremia. And what we've actually found is there are individuals who have been exposed as many as 6 times to hepatitis C. We detect 6 different hepatitis C viruses, and they've cleared them all. So I would call these guys the super HCV controllers. And I think we really need to learn, as Nagla Shukri and other people are doing, with us trying to learn more about how these individuals control Hep C over and over and over again. So that tells you that there's at least some evidence for natural protective immunity. So can we actually mimic that through vaccination? And there are some significant challenges to the development of a Hep C vaccine, and they're in many ways quite parallel to the challenges we see in trying to make an HIV vaccine. These are really highly diverse viruses. Hep C even more than HIV. These are phylogenetic trees of our 3 most common chronic infections. at least transmissible through blood, common infections, hepatitis B virus, HIV, and hepatitis C. These are drawn on the same scale. So HIV is famous for being an incredibly diverse virus, but truthfully, hepatitis C is about an order of magnitude more diverse. So when you say I'm going to make a vaccine against hepatitis C, the question is which hepatitis C, because it's quite diverse. Traditional vaccine development has been done using live attenuated pathogens or killed pathogens. There is always a small risk of infection in those circumstances, which makes this fairly untenable for HIV or Hep C vaccine development. In addition, it's very hard to culture hepatitis C, and so the notion of growing it up and passaging it or creating genetic mutations that attenuate it or growing it up enough that we can make large batches and kill it really doesn't work. So this is not a feasible strategy, and we have to think about more modern strategies. So in addition to the really pioneering work done by Michael Houghton, looking at induction of antibody responses against hepatitis C and trying to generate a protective response by inducing antibodies early that prevent infection, there's an idea that we may also need to invoke an excellent T-cell response, and this has been propagated in both HIV and hepatitis C, and I'll point out in Ebola and some other pathogens as well. And so why do we think T cells are important? Well, there's substantial evidence that T cells are crucial in control of hepatitis C. When we look at genetic polymorphisms associated with control of hepatitis C, in addition to some innate sensing genes, the lambda interferons, we see associations between human leukocyte antigens. And so those are the things that present peptides from Hep C to T cells. And there are associations in HIV and Hep C with control based on a person's HLA type. Chris Walker's group with Nagala Shukri and Arash Grakui depleted T-cell populations from chimpanzees and exposed them to hepatitis C, and those studies very compellingly demonstrate that T-cells are critical in controlling this infection. In addition, there have been associations with the breadth and magnitude of T-cell responses with viral clearance. And we've seen the induction of interferon gamma, which is an antiviral cytokine produced by Hep C specific T cells exactly around the time that there is a reduction in HCV RNA in at least a few studies. So we think that T cells are important. How do we get good T cell responses? One of the modern methods for vaccine development that has not yet been put into a licensed vaccine but has been tested in a variety of settings and is moving forward is to use vectors. These are other pathogens with mutations that render them fairly non-pathogenic, and they're fairly non-pathogenic to begin with, that carry antigens of a virus we care about, hepatitis C, and that is designed to induce both innate and adaptive immune responses against the virus. So the problem with this is these viruses or bacteria that we use to stimulate these immune responses are often viruses to which we're exposed. And the reason is that we pick them because they're fairly non-pathogenic, so they don't kill anybody, and we all often tend to be infected with them and do just fine. But the problem is that we sometimes also already have immune responses that just clear the vector so quickly you can't make a response to the vaccine antigen. So there is a company, Okairos, that actually searched for adenoviral strains that didn't have, that in non-human primates worldwide, and the goal was really to discover adenoviral vectors that are highly immunogenic, so induce really potent immune responses, are easily manufactured to high titers, but don't stimulate cross-reactivity, so they don't get cleared quickly by the immune responses we've already generated against similar viruses. And so they were actually successful in doing this. And what they did is they isolated over 100 novel adenoviruses from chimpanzees, and they basically found that this is an effective source of viruses because the hexon proteins of adenoviruses that circulate in chimpanzees differ from the hexon or surface proteins on our adenoviruses that infect us. So there actually wasn't much cross-reactivity, and they found many that were very highly immunogenic. So the vaccine strategy they employed was to use a low seroprevalence chimpanzee and human-derived adenovirus. Really, it's this chimpanzee adenovirus 3 that they've moved forward. So this comes from chimpanzees, and doesn't induce cross-reactive immunity. And then they used that's the first vaccine. For the second dose, that's the prime. For the second dose, or the boost, they actually decided to use modified Vaccinia Ankara, which was given to hundreds of thousands of people worldwide previously in a vaccine campaign to eradicate smallpox. So this is a second vector system that's widely used in vaccine design. And those are the carriers. What do we use for the antigen? How do we make a Hep C specific response? The answer is that we take the non-structural regions of the protein. That's the vector now have the non-structural regions inside. And the idea is that this will make the proteins, the non-structural proteins of hepatitis C and T cell responses against them. There are many CD4 and CD8 T cell epitopes contained within this region of HCV, and there's some conservation of those epitopes. And this vaccine was developed in Europe, and as a result, the genotype of the virus, the protein that's in there, is actually genotype 1b. So the aim is really to induce antiviral immunity with functional characteristics associated with viral control and natural infection. And that essentially is broadly targeted, durable, functional CD4 and CD8 T cell responses. So how does this work? This was first tested, as you can imagine, in healthy volunteers and not in at-risk populations. And it's a highly potent inducer of T cell responses. All individuals who received this vaccine made a response, and many of them developed responses against multiple HCV proteins. And so shown here those are the proteins contained within the vaccine on the bottom. And on the y-axis is the amount of interferon gamma being produced by cells from these patients. And so the higher, the better. And for the immunologists, look at the axis, because these are huge T cell responses, and against really multiple different proteins within hepatitis C. In addition, when they assessed those T cells, they made many cytokines that have been previously associated with control of other infections when vaccines have induced those responses. And one question is this going to work across multiple genotypes of hepatitis C? So if we look here, again, same scale on the Y, now we're looking at responses to different genotypes. Probably not surprisingly, the responses are most robust against genotype 1 hep C peptides, but you can see that there is some response to even genotype 3A or 4B peptides. So there is some cross-genotypic responding going on here. It's just not as robust as the response to genotype 1. And the vaccines were shown to be very well tolerated and safe in this healthy population. So with that, we moved it into an at-risk population in a trial called Vaccine as Protection, or VIP. It's a double-blind, randomized, placebo-controlled trial that was initially designed by Kim Page and by me to take place at Johns Hopkins in Baltimore, and the University of California, San Francisco. Subsequently, the University of New Mexico became involved because Kim Page moved there. So the population we're enrolling are 18 to 45-year-old people who are actively injecting drugs and who are not infected with Hep C at the time we encounter them. And of course, we provide them with extensive counseling about the risks of drug use, how to keep themselves safe, refer them to needle exchange programs and drug treatment programs. The size of this trial is will be about 540 people at closure, and the goal is to assess the safety, the induction of immune responses, and the ability to prevent chronic infection. So this is key. We're not aiming for sterilizing immunity in which people don't become infected, but we're hoping that no one stays persistently infected, and the reason is that that's really where we see disease for the most part. So this is 2 vaccinations, as I mentioned, starting with this chimpanzee adenovirus and then modified Vaccinia Ankara as a boost. They are separated by 8 weeks, and we are assessing immune responses at multiple time points throughout the trial. We test them for HCV RNA on a monthly basis to determine if they've become infected. And so in conclusion, I would say that we do still need a prophylactic vaccine for hepatitis C virus infection. And sorry, I probably should have said this, but I'll say in the conclusions that the results of that trial should be available later this year. There is probably protective immunity in vivo, and our goal is really to mimic that with a vaccine. As with HIV infection, it's going to be very challenging to make a vaccine that protects against the diverse strains of hepatitis C virus, but We do have one for the first time in testing with results expected later this year. And an important point is I think this shows that it is possible to use this patient population as a vaccine test population. And with that, I'd like to acknowledge on the left, basically the University of New Mexico, Hopkins, and UCSF are study staff. I showed a lot of data generated in healthy volunteers. Those data really come from the Oxford team testing these vaccines. The vaccine was developed with BioKairos, which was subsequently purchased by GlaxoSmithKline, and all of this work was funded by the United States National Institutes of Health, and central, and that's why they're in the middle to all of this, are our study subjects. Without them, I couldn't have shown you what we've learned about natural protective immunity, and without them, we'd have no ability to test whether this vaccine is protective. So, thanks. Do the questions now? Questions now or later? Now. Okay. Yeah, thank you very much for your attention. I'd be happy to take any questions.
Is there a microphone going around?
I can hear you. And I can repeat the question. Yes, so the question was, are the people who became persistently infected with HIV in the B-BASH cohort? Actually, the data I showed, because we didn't want to confuse the matter, were from HIV uninfected individuals, all of them. We do have a patient who acquired hepatitis C, cleared it, acquired HIV and hep C, and became persistently infected, but I showed data only from individuals who are HIV uninfected. But there's no doubt that having HIV is a risk factor for persistence of hepatitis C, and that wouldn't be surprising.
Forgive me if I missed it, but it looked like you only put in non-structural proteins?
Yeah. I didn't make the vaccine, but so.
I would have done it anyway.
So the vaccine, you're correct, only contains non-structural regions and would therefore not be predicted to induce neutralizing antibody responses. Whether you get a modification of neutralizing antibody responses is an important scientific question we're going to assess, but it would all be through T-cell help and not through a response to the antigen of the vaccine.
Yeah, I mean, obviously the T cells for sure, but I would think you'd want to have both.
Yeah, I agree. I agree, and as I said, I have no financial stake in this vaccine, but I made that point early on, and for complicated reasons, it was difficult from a manufacturing standpoint for them to get that in. So one thing that's being considered is perhaps using the vaccine that was developed, yes, at Chiron by Michael Houghton, to see if we couldn't perhaps combine the responses that are inducers of potent T-cell responses and induce good neutralizing antibody responses. But we need to see what the results of this trial are before we know whether we need to make improvements or not. But that is one potential area of improvement if we don't see the response we hope to see.
Thanks for a nice talk.
Thank you.
I was just wondering, in the groups that are being tested for these T-cell response vaccines, are they limited to uninfected individuals and those who have spontaneously cleared? And if so, would you expect the same sort of T-cell response profile in individuals who have been treated with their chronic infection?
Yes, so the individuals enrolled in the vaccine prevention trial have to be hep C antibody and RNA negative, as well as HIV and hepatitis B uninfected, and some other things that modulate responses to vaccine and also to hep C infection. So in fact, the vaccine has been tested in individuals who are chronically infected, and the T-cell responses induced are to targets that are not contained within the person's circulating virus. So there's no doubt it's not effective as a mechanism of controlling hep C, but I think we could argue that that's not necessary given the DAAs. So there's no doubt that pre-exposure to hepatitis C could potentially modulate the response. And that may have implications not so much in treating chronic infection, but I mentioned that reinfection is a problem. So this, as soon as we get an effective vaccine, and perhaps even beforehand, we might begin testing in people who've been treated with DAAs to see if we can reduce the rate of reinfection. It'd have to be a very special target population that's at risk of reinfection.
Thank you.
Thank you for a great talk. You make working in a challenging population sound very easy. But can you give us a sense of how tough it is? So in your cohort where you're bringing them back every month for testing, do you have a sense of what your dropout rate is and what is your mortality rate in your duration of follow-up?
Yes, so you sound like someone who's had some exposure to this population. There's no doubt that this is an enormous challenge, and my hat's off to Julie Bruneau and others here who have really created similar cohorts, because this is not the easiest patient population to follow. And so for obvious reasons, the vaccine was tested first in healthy volunteers, in which there was no chance of assessing efficacy, just induction of immune responses and safety. So moving into this population has had its challenges. There are more severe adverse events than there would be in a healthy volunteer trial, but we're pretty sure that a stab wound is not caused by the vaccine. So usually the adverse events are fairly separable from things we think might be caused by the vaccine, but there's no doubt we've not had a lot of mortality, fortunately, but as you may know, we're in the middle of an opioid and heroin epidemic in the United States, with overdose being the leading cause of accidental death in 2014 over car accidents. So overdose is something that we worry about, but all of our participants are instructed in the use of naloxone to reverse opiate overdose. So it's challenging to get them to come back, but we found that in addition to offering them incentives that are more obvious, like money, cards for food, those kinds of things, that really providing them access to these services is another benefit. So many of them come to us when bad things happen to them because they rely on us to fix all kinds of problems. And that really creates a long-term relationship with them that enables us to help them, not just through the vaccine trial, but to really get them on a different path in life in some cases. So that's a big benefit to them, and that is something that they really seem to like. Any other questions? Okay, thanks again.